we'll talk about landscapes in CRISPR binding. Yeah, go ahead.
Okay, you can see it, right?
Yes.
Okay, hi everyone. My name's Pete Regas, and I'm a graduate student in the Lambert Lab from Cornell University. And the talk that I'm going to give is titled Computation of Exit Times for Potential Landscapes in CRISPR Binding. So to begin, I just wanted to talk about binding thermodynamics. The particular CRISPR-Cas protein that we're going to be considering is FNCas12a. This protein has target sequences that are 20 base pairs long. And the inspection process for this protein entails that it first recognizes a PAM sequence, then search through a target sequence on the DNA for complementarity, from which a stable nucleus is formed. And from this figure here, we can see that this is an example of such a CRISPR system in which the protein is diffusing in one dimension along the DNA strand throughout its complementarity search. And for an introduction, we're going to be looking at the exit or passage time distributions tau of x in connection with the figure that I gave on the previous slide. The connection is that once we look at whether a passage time has occurred to an absorbing membrane, say of length 1, 2, and 3, and so on, this will actually indicate that the complementarity search for the protein, in particular for Cas12a, is undergoing, and that the nucleus can actually stably form. And the differential equation that we have to model this relationship is a second order dimensionless differential equation. We've normalized it in a particular way such that there's nothing in front of the second derivative term, but the first derivative term and the constant are normalized by the inverse of the Boltzmann constant and the temperature. But without a doubt, the most important part in the component is this landscape term u prime of x. And we're solving the initial value problem according to these conditions. And we can visualize that within some plasmid, the steps of the landscape barrier that have to be overcome is, first of all, the n is equal to 0 step, which involves the PAM inspection. And then for the n is equal to 1 step, the protein, while it's still inside the plasmid, it will begin to inspect the remaining parts of the sequence that's given in this other color. And then after following a finite number of steps in the landscape, the stably bound complex will be formed. And for the goals of our talk, one very useful figure to have in mind is this figure, which captures how the reconfiguration rate differs depending on a base pair mismatch in the sequence. The reconfiguration rate is significant for the formation of the stably bound complex because it describes the energy barrier that the protein must subsume in this reaction. And we can see that for the 20 base pairs of the target sequences for FNCas12a, depending on the base pair mismatch location, there's a variable reconfiguration rate that we would expect from statistical mechanics and thermodynamics. And so besides one of the broader goals of simulating the exit time dynamics for the target sequences, we want to verify specific predictions that we have for Cas12a, in which, as we can see in this first region within the first 6 base pairs, when a base pair mismatch happens, this drastically affects the reconfiguration rate. While in region 2, we can look at base pair mismatches for the remaining parts of the sequence before the last 6 base pairs, in which there is still an effect, but it's not as significant. While for region 3 poses very little impact on the formation of this stably bound complex. And also, I will illustrate how, from the analytical solution that we can obtain from this initial value problem, how we can obtain a temperature dependence on the binding process from our approximations. And so, for this specific class of potentials that we'll be looking at the Dirac delta potentials. For our purposes, this is the most realistic because this class of potentials has discrete energy barriers that the protein must overcome in the binding process. And for this simplified potential, the solution for tau of x takes this term, which involves a linear term, an exponential term, and a constant term that are all modulated by either the absolute value or the square of the absolute value of the magnitude of the jump of the landscape potential at base pair of inspection x sub 1. And this solution will look very similar to the other solution that we will manipulate later on. And so to work towards our general goal of simulating exit time dynamics, what we'll look at is we'll take a typical delta landscape such as this one. The potential or energy barrier can either be constant across base pairs or it can be variable. But what we'll do is that we'll prepare Gaussian distributions over each neighboring base pairs, and we will derive a dependence in our analytical solution for the height and the variance of the distribution, from which we can perform several computations and solve these initial value problems in parallel. So step 0 and 1 to construct such distributions just includes setting the distribution very close to the base pair of inspection, and for that's step 0. And for step 1, if we look at the PMF for a normal random variable and shift it upwards of h units, we can obtain an expression for the PMF which can serve as a distribution, given that the height h is equal to the difference in the landscape potential at base pairs of inspection I plus 1 and I. And then for the remaining steps 2 and 3, we can numerically approximate the exit times given these initial conditions in the potential, in the Gaussian potentials that I've constructed. And conveniently for the Gaussian potential or the Gaussian distribution, the supremum of the probability mass function across each base pair over which we construct it is approximately given as 0.4 divided by the variance. And then for the final step, we'll collect the approximations from our simulations. So analytically, this expression is where we actually gain a lot of traction and where we're able to perform several of these computations altogether. The analytical solution for the initial value problem takes the form as the inverse of the Boltzmann constant times the temperature multiplied by an n squared term, an x squared term, and this double integral term, where we approximate the orange term with a trapezoidal sum, and where in each part of the solution, the function f that we're integrating against is an exponential whose power, which is a prefactor to the error function here, is dependent on the variance of the distribution and inversely proportional to the Boltzmann constant in the temperature. And conveniently for us, we can decompose the solution in this way so that we can avoid some issues with actually having to restore initial conditions for the value problem after each iteration. And from the analytical solution that we obtain in the exit time, we can obtain a very clear expression for actually the exit time fluctuation in between neighboring base pairs of inspection, which takes this form. Thank you. And from f of u, we can observe that the power of the exponent roughly takes this form, and this is a trapezoidal sum approximation, and this is the initial and final values of the height that I use, and that's the value that I introduced for a landscape barrier fluctuation, and these are the temperature values. So for the first simulation, what we're able to achieve is that we're able to simulate for approximately 400 base pair sequences. We're able to observe a substantial change in the magnitude of the exit time that we approximate, and we are able to clearly obtain a temperature dependence in which we run the approximation for 8 temperature level sets. And with this value of the parameters, we can also provide and shed light on the remaining predictions that we have for regions 1, 2, and 3, because we can see that depending on the location or the base pair mismatch, we can observe either substantial changes in the predicted exit time that we have for this reaction process, which are specific to FNCAS12A. And as a conclusion, I introduced in my talk the binding dynamics, I provided analytical solutions, and I also described the formulation in which we obtain analytical approximations that I'm hoping to apply to additional CRISPR-Cas proteins from my colleagues. And I wanted to thank everybody for their attention in my lab, and if there are any remarks or questions.
Yeah, we have time for questions. There was a question, how much does the thermodynamics of binding vary between different Cas systems?
Yeah, it just depends on the actual base pair of inspection. So, and the actual temperature and duration of time during which the stable complex is formed. So we're able, in our approach, we're able to accommodate for all of those factors, and even in the simulations that I provided for different temperatures and different base pair sequences and different timescales. So that's what differs.
And how do your results relate to the accuracy of using CRISPR for modifying genomes?
Yeah, it relates to the accuracy because usually when we're targeting the genome and we have this CRISPR-Cas protein, as I mentioned, as it inspects the PAM sequence and the target sequence and binds, we usually measure the change of the rate in the transcription factor by the difference of fluorescence in gene circuits. So it can help in that respects just by how we can use those experimental measures, which again, my colleagues are doing in the lab, but that's the efficacy of either blocking a gene, completely knocking it down, or having fluorescence for its expression.
Okay, thank you.
Okay.
Thank you
Thank
very
you.
much. Then please, yeah. And then we are ready for our next speaker. That is